Welcome, ladies and gentlemen. After a long break from videos, we're going to talk about cell communication. This is just going to be kind of a general introduction to the topic, and then we'll be looking at more specifics in the next lesson. So let's talk about communication just in general. Communication, whether it's between an organism, whether it's between cells within an organism, whether it's between an organism and a single-celled organism and another, occurs in three stages. You have some sort of stimulus, something that carries a message, like for example, what I'm showing you here is a stoplight. So that green light is a signal, okay, traffic can move through. But then when it turns from yellow to red, any drivers who are on the road will integrate that signal, in this case in their brain, and then they will have an appropriate response. In this case, their response is to put the brakes on and stop at the stoplight. We're going to look at this theme fairly regularly throughout much of what we're doing in this unit. We're going to look at what is the stimulus, where is it integrated, and what is the response. So cell communication happens on multiple levels. Organisms that are unicellular can react to their environment and can react to nearby organisms through these kind of cell communication molecules, and multicellular organisms have communication between their individual cells through these same mechanisms. So cell communication is mostly a matter of chemicals being exchanged. Chemical messengers are used to basically indicate that a stimulus has been received, and then they will set in motion a series of actions that will produce a response. So there has to be some way to get a signal from outside the cell to the inside of the cell, and usually this is through the interaction of two molecules, a ligand or ligand, either way is proper to pronounce it, and a receptor. A ligand can be any molecule that acts as a signaling molecule. The only restriction on it is just that it needs to bind to a specific receptor. A receptor is going to be a protein, most likely a protein. Very many of them tend to be located embedded within the cell membrane, like the one that you see in this image right here, where we're looking at a ligand outside of a cell which has a receptor stuck in its membrane. When a ligand binds, that causes a shape change in the receptor, which then causes it to trigger a change in something on the inside of the cell. And each receptor has a very specific ligand. So if you don't have a receptor for a particular ligand, you could put a ton of that ligand outside of that cell and it won't respond. That would be like somebody coming up to you, and if you don't know sign language, talking to you in sign language. You don't understand that signal, so you wouldn't have an appropriate response. Or it would be like a bat coming up to you and trying to communicate with you by using frequencies that are too high for you to hear. You don't have a receptor for that, so you can't receive that messaging. So let's take a look at this happening. A ligand has a very particular fit with a receptor, just like an enzyme has a very particular fit with its substrate. When it binds, that's usually going to cause some sort of conformational change in the receptor. In this case, our receptor has lost affinity for that star-shaped molecule, and that star-shaped molecule then went within the cell and ended up triggering a signal of some sort. So we'll talk about ligands, we'll talk about receptors throughout at least the first few PowerPoints of this unit. All right, so there are two major ways we can break down cell communication. There's the short distance type, and then there's the long distance type. There are two different short distance types. Actually, there's going to be a third one that we'll look at soon, but there's autocrine and paracrine signaling, which is going to be very short distance, and those molecules that are acting as signal molecules are only going to travel by diffusion. Long distance signaling is called endocrine signaling, and that is usually going to involve molecules that are carried by some sort of circulatory system. In humans, for example, they're carried through the blood. If they were being carried around through a plant, they would be carried through its vascular tissue, the xylem and the phloem. And these chemical messengers for endocrine signaling are called hormones. We'll talk about that more in a few minutes. So there are a couple of different terms. I just wanted to throw this term at you in case it shows up on a test. Juxtacrine signaling is a term for two cells that are physically connected. I'll show you an example of that soon. Paracrine signaling is neighboring cells, so they don't have a physical interconnection between them. So this is juxtacrine signaling. Apologies, forgot to put things on silent. So juxtacrine signaling involves having molecules move directly from one cell to another without having to cross a cell membrane. This means that you can allow all sorts of molecules. They can be hydrophobic, hydrophilic, and they don't have to cross through the boundary of a cell membrane. There are certain molecules that this is more common with. Growth factors can do this. Some cells that mediate the immune response can also communicate this way, and plants will often communicate with cells within plants will communicate this way. There are different types of junctions in animal cells that allow this type of communication. They're called gap junctions. And then within plants, they're called plasmodesmata, which are basically these little channels that go through the cell membrane and cell wall and carry a little bit of cytoplasm in there. One of the things we'll talk about in our next unit when we talk about DNA is the fact that you can also have small RNAs that pass from cell to cell. Viral DNA can also pass from cell to cell this way too. Cells can also have basically cell-to-cell surface recognition. So there are proteins on the surface of the cell. They interact, and then that sends a message. This is really common. Our biggest example is going to be the immune system. So let's look at an example. Here we have a cell. It's got a Y-shaped receptor on the surface, and that Y-shaped receptor has that teardrop-shaped molecule attached to it. Another molecule comes that has a ligand that fits with that Y-shaped receptor. That serves as a stimulus, which then causes that Y-shaped receptor to change shape so that the teardrop-shaped molecule moves away. Notice it's moving into the nucleus. It's doing something. We'll go into more detail about what's happening in future lessons, but it's making some RNA. That RNA goes out, is transcribed, and used to make molecules that are released from the cell. So here you see all three stages: stimulus, integration, and response. So one cell received a stimulus from another cell, and that caused it to respond by producing a certain molecule. This is actually the mechanism by which some of your immune cells are activated to produce antibodies, which are what these Y-shaped molecules are. So, like I said, immune cells have this direct interaction between molecules on their on their surface. All of our cells have these markers on the surface called major histocompatibility complexes, or MHCs, and these are the way that your body identifies what cells belong as part of you, and what cells are a part of the body versus cells that are foreign, cells that are invaders. So this is called identifying self versus non-self. So these molecules are found on the surface of the cell, and immune cells will come along and they will have interaction with those receptors. If they have the appropriate MHC molecules, then no reaction. If they have some sort of foreign material on them, that will trigger a T cell to start dividing and start creating antibodies, recruit other cells, etc., and that will then trigger an immune response so that your immune system can fight off whatever's infecting you. These cells that are presenting these fragments of molecules on their surface are called antigen-presenting cells. Notice what happens is, let's say you have a microbe here, and notice the surface proteins are kind of broken up, and within this immune cell, some of those surface proteins are attached to one of these MHC molecules, and that is used to kind of basically train a T cell, hey, this is what you're looking for, this is the infectious agent. So let's talk about another type of local signaling. Paracrine signaling, like I said, neighboring cells. You're going to have signaling molecules that are released into the surrounding fluid, whatever it is, and then they will diffuse to their neighbor. There's a very specific type of paracrine signaling called synaptic signaling, where nerve cells communicate with nearby nerve cells. So we may have talked about, or we may talk about in the future, about how signals are sent along an individual nerve cell. That is an electrical signal, but between nerve cells, that signal changes form. It becomes a chemical signal, and that chemical signal involves the release of molecules called neurotransmitters, which act as ligands to receptors on a target cell. And that's going to cause some sort of response in the target cell. If the target cell is a neuron, it might cause it to send another signal. If the target cell is a muscle, it might signal it to contract. If the target cell is a gland, it might signal it, hey, release some hormones. Just kind of depends on what the target cell is. So just a close-up of synaptic transmission. You have, honestly, sitting around in the ends, or what's called a synaptic terminal of neurons, are these little vesicles, bubbles basically, that are bags of membrane that contain neurotransmitters. They're sitting around waiting until a signal, a nerve impulse, comes down to the synaptic terminal. Once that happens, that triggers the fusion of these synaptic vesicles to the cell membrane, and that exocytoses or secretes their contents into the gap between the nerve cell and its target cell, which is called the synaptic cleft or the synaptic gap. It's a very short distance, so molecules can diffuse across very quickly. They will interact with a receptor, and then they will trigger some sort of response in the cell. Then those neurotransmitters are either very quickly taken up again by the original neuron, or they're destroyed. So that way there's a very short, specific response. Growth factors also act as paracrine signalers. So a growth factor is, like its name says, something that encourages cells to grow. It basically encourages cell division so that you'll get more of a specific type of cell. These growth factors are really essential in development during embryogenesis. So when you were developing from that single fertilized egg cell to a baby that was ready to be born, there was a very complex interplay of growth factors that were being released inside of your body. As adults, growth factors also play a role. Unfortunately, they can also play a negative role. For instance, tumor cells often release growth factors. When you see this GF at the end of the name of a growth factor, that literally stands for growth factor. So this tumor cell is releasing growth factors that are stimulating it to divide. That's autocrine signaling, which you have highlighted here. And then paracrine signaling is that it will then trigger nearby cells to divide as well. One of the ways that this is beneficial to a tumor cell is it can encourage blood vessels to grow towards it, so that way it receives excess nutrients and also a plentiful supply of oxygen. Obviously, that is not the ideal condition, because then that tumor is going to be able to grow more quickly, and then you'll have cancer and it will be unpleasant. As far as the normal role of these growth factors, there are these molecules called morphogens. Morph literally means form or shape. Gen means create. Like the genesis of something is the origin or the creation or the beginning of it. So what will happen is some cell in a developing embryo will be responsible for releasing a molecule called a morphogen. There are a number of different morphogens. They are very important signaling molecules that we'll talk about a little more when we talk about DNA. But based on the concentration of this morphogen a cell is exposed to, that will determine what type of cell it becomes, whether it becomes a nerve cell, whether it becomes a skin cell. I mean, I'm oversimplifying this, but these morphogens are very important for determining, hey, what's going to be a finger, what's going to be a thumb? what's going to be your pinky, what's going to be a middle finger. And so your cell identity is determined by the concentration of these morphogens. So I really like this bottom figure because it shows a cell that is producing a morphogen. And you also have a graph at the bottom, which is representing the cell on kind of the x-axis, distance from the morphogen producing cell, and here is the concentration of the morphogen on the y-axis. So notice right next to the morphogen producing cell, you have a high concentration of morphogen. That will trigger those cells to differentiate into a specific type of cell. Farther away, you're going to have lower concentrations of that morphogen, which will indicate a different cell fate. It's really complex, but really kind of neat when you start to look at it. Another way in which cell signaling is autocrine and paracrine is a phenomenon called quorum sensing in bacteria. So a quorum is like the minimum you need to do something. So if you're doing like some sort of government function, like you have like a session of, the Senate or session of, the state legislature, they might have to have a quorum, which is a certain minimum number of members there that have to be there in order for you to be able to vote on anything. Quorum sensing occurs when bacteria start to build up in concentration. So bacteria are all individual single-celled creatures. However, when enough of them are found in a small area, when there's a high enough concentration of them, they will actually start to change their behavior and cooperate with each other to form these structures called biofilms. They're kind of goopy. They're probably not very fun to touch. And they have the cells that are in these biofilms are going to be producing different molecules and acting differently than cells that are kind of spread out. So what happens is bacteria are constantly releasing these autoinducer molecules that they themselves have a receptor for. So that's autocrine signaling, and they are capable of responding to it. The higher the concentration, the bigger the response. However, if it's just a single bacterium swimming lonely in some liquid somewhere, it's not going to have a lot of autoinducer molecule out there, so it's not going to change its behavior. Meanwhile, that bacterium starts dividing and starts dividing and starts dividing, and the concentration increases. Then what is going to happen is there's going to be a higher concentration of that autoinducer. More autoinducer receptors are going to have this autoinducer bound to them. So for example, this guy right here has 4 receptors, all with an autoinducer bound, which is going to trigger some changes in gene expression, and that bacterium will start to produce different proteins, which will make it behave differently. And so this allows these bacteria to cooperate with each other and enhances their survival because they're cooperating. They're not like they're working together for a common goal, which means they all have a higher chance of survival. So now let's look at long-distance communication. That's endocrine signaling. You have these signaling molecules called hormones. They are released into body fluid of some sort in us and other animals. That's usually in the blood. Like I said, in plants, that's going to be in their in their vasculature, their vessels. And then it travels through the blood vessels. It ends up getting spread to the entire body, but only the cells that have a particular receptor will respond. So you have, millions of cells in your body, maybe 1,000 of them might have a receptor for this particular hormone. And they are the only 1,000 cells that will change their behavior in response to this hormone. So you can have a bunch of hormones released in your blood. All of your cells get exposed to it, but depending on whether or not you have a receptor for it and what that receptor does, individual cell types might have different responses, which is why you can have, for example, adrenaline, which is a hormone. It's the one responsible for your flight or fight reflex. When adrenaline is released into your blood, it has different impacts on different parts of the body. So it speeds up your heartbeat, it speeds up your respiration so that you can have plenty of oxygen and be able to pump blood and the oxygen and glucose in it to a lot of different areas in the cell. in the body. However, it has the opposite effect on the intestines. It slows down digestion and diverts blood away from your intestines, so that way it can be used to help power your muscles. So they have different receptors, intestinal cells versus heart muscle cells, and so they'll have different responses. There are a number of different classes of hormones. I have not seen anything on the AP test that would require you to know the difference between any of them. The only thing I want to point out is you can have hormones of various types. There are a lot of protein hormones. However, there are also very important steroid hormones. In fact, these are the sex hormones. They are made ultimately from cholesterol, and they have that same kind of fused ring structure that cholesterol does. They are hydrophobic. They are capable of passing through cell membranes, whereas proteins, especially large proteins like human growth hormone that you see here, are not hydrophobic, cannot pass through the cell membrane, and therefore have to interact with a receptor somewhere on the surface. You also have hormones that are found in plants. We'll do our best to talk about a couple of examples of those, just so you can learn how plant cells respond to hormones as well. And like I said, they're very specific. Even though the entire body is exposed to them, only certain cells are equipped to actually respond to them, so they only affect a particular set of cells. So other examples, there's insulin, human growth hormone. There are a lot of hormones released by your thyroid, and then the sex hormones like testosterone, estrogen, androgens, progesterone. All of those are examples of hormones. So let's look at testosterone and estrogen production. So depending on which sex organs you have, whether you have ovaries or whether you have testicles, you're going to produce different sex hormones. If you are female and you have ovaries, they will produce estrogen in higher quantities than testosterone, though I will tell you, both males and females produce both of the secondary sex hormones, testosterone and estrogen. And then if you are male, the testicles will release more testosterone than estrogen. This will be released into the bloodstream. The bloodstream has access to the entire body, where it will eventually reach the pituitary gland. When the pituitary gland receives those signals, it will then release its own hormones in response, which can then affect other organs. Another example of this is insulin, and we'll talk about this a little more, especially because we're going to talk a little about how insulin actually has impacts on the cellular level, because it's a good example of messaging within a cell. So you have a very complicated system to keep your blood sugar in optimal levels. This is maintaining your blood sugar homeostasis. We'll talk about this pathway in more detail later, also because we're going to be talking about feedback. So what ends up happening is, let's say that your blood sugar is low. If your blood sugar is low, then what that's going to do is that's going to stimulate your pancreas to produce glucagon. Glucagon travels throughout the bloodstream and it has impacts in multiple tissues, but we're going to focus on the fact that when it reaches the liver, it stimulates the breakdown of glycogen and produces more glucose. That glucose gets released into the bloodstream, raises your blood sugar. Once your blood sugar is higher, that's going to block production of glucagon, so you're no longer releasing more sugar into the blood. Let's say that your blood sugar is too high. In that case, your pancreas is going to produce insulin. Insulin has impacts on all of your tissues. It also has impacts on the liver. So in the liver, it basically says, hey, take in glucose, make glycogen, store this glucose, we've got too much. It also triggers all your tissues to start taking in glucose. That's why if you have diabetes, for example, your body either doesn't make insulin or doesn't respond to it, and that's why you have trouble maintaining a proper blood sugar, and that's responsible for all the health effects there. So that is it for now. Be prepared for a little more complication in the next slide.